Chapter 2, Part 2, Biochemistry. Biochemistry is a study of the chemical composition and the reactions of living matter. In biochemistry, we're going to look at two main groups of compounds. The first of those, organic compounds. These contain carbon, are covalently bonded, are usually very large molecules. The inorganic compounds that we will discuss usually do not contain carbon. The three main types of inorganic compounds we will discuss are waters, salts, and most acids and bases. We will begin discussing inorganic compounds. The first of these inorganic compounds that will be important is water. Now, we all know that water is important for life, and today we'll discuss some of those reasons why water is so important. Water has certain properties, the first of which is a high heat capacity. This is the ability to absorb and release large amounts of water before changing temperature. This allows water to act as a coolant within the body, redistributing heat and helping to maintain homeostasis. Water also has a high heat of vaporization. This means it can absorb a large amount of heat before it actually changes from a liquid to a gas. This is another way in which water helps to cool the body, and what we see here is that sweating, the process of creating liquid water on the skin, which then takes heat away as it changes into a gas and evaporates. The third property of water that's important is that it acts as a polar solvent. Because water is a polar covalent molecule, as discussed in the previous lecture, we will see that it can dissolve ionic substances, specifically things like sodium chloride. So here we have sodium and chloride, an ionic molecule. When we take something like salt, seen here, and put it into water, the polar molecule of water will actually help to dissociate these ionic bonds. This allows water to serve as the major transport medium of the body. Two of the other properties of water include reactivity, in which we will see throughout today's chapter the idea of hydrolysis, which is the breaking of bonds by the addition of a molecule of water, or dehydration synthesis, which is the creation of bonds through removal of a water molecule. The last part then is cushioning, and if you think about a waterbed, water is resistant or resilient, so we see water acting as a cushion around certain body organs. The second type of inorganic molecule are salts. These contain cations other than the H+, or the hydrogen ion, and anions other than OH, or the hydroxyl or hydroxide ion. These act as electrolytes, and they allow for the conduction of electrical currents. For example, here we have NaCl, or sodium chloride. This is our typical table salt. When mixed in water, we would get sodium ion and chloride ion. Here we have calcium carbonate. Calcium carbonate is another example of a salt. When mixed in water, we would get the calcium ion, which is Ca2+, and this is the carbonate ion, which is a negative or anion. Finally, the last example given here is potassium chloride. Potassium chloride is functionally very similar to sodium chloride, and when mixed in water, we would get the potassium ion and then the chloride ion. Now, since we have specified that salts contain cations other than H+ and anions other than OH-, that suggests that those two things are special, and indeed they are. These are the formation of acids and bases. So acids release H+, and we refer to these as proton donors. If you think about the form of hydrogen, hydrogen in its atomic form has one proton and one electron. So if it loses an electron to become an H+ ion, all that's left is a proton, hence the term proton donor. Bases, on the other hand, release OH- or the hydroxide ion, which is then called a proton acceptor. Now, in terms of acids and bases, we measure these based upon the concentration of the H+ or the proton. We use a pH scale as a measure of acid-base concentration. This is a measure of the number of hydroxide or hydrogen ions within a solution. It is a logarithmic scale, so as we look at the pH going from 0 to 1, 1 to 2, we actually see an increase of tenfold, right? Hence it is a logarithmic scale. We see acidic or acid solutions have a pH that is in the low numbers, so over here going from 0 to 6.99 would be acidic. Alkaline, shown in blue, would be over here, 7.01 up to 14. Perfectly neutral solutions such as distilled water would have a pH of 7. In a pH of 7, the number of hydrogen ions and the number of hydroxide ions is equal. As we move to organic compounds, we will see that organic compounds are unique to living systems and they contain carbon. There are 4 main categories of organic compounds we will briefly discuss. The first are carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen. The examples we will see are monosaccharides, disaccharides, and polysaccharides. Monosaccharides, meaning one sugar. Here we have a simple organization where we have a ring-like organization of carbons. This is a supply or a source of cellular food. Some of the examples we will see are glucose, fructose, galactose, deoxyribose, and ribose. Notice along this ring structure, here, 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 these points are where we have an implied carbon that you cannot see, but it is there. Glucose is the preferred monosaccharide for cells. Okay, so this is what cells want to use as their energy source. In terms of sugars, though, what we think of as sugars are actually disaccharides. So most of us don't put glucose on our cereal in the morning. Instead, we put table sugar or sucrose. Sucrose is actually a disaccharide, so 2 of these single molecules held together with a bond. It is actually a glucose and a fructose together. Now we put the glucose and fructose together through dehydration synthesis, so you'll notice an OH here and an H here. If we were to pull those out, we would create a bond between these 2 molecules, and here we see our first example of dehydration synthesis. In breaking a bond in sucrose, we can use what's called hydrolysis, breaking using water. Using a molecule of water, we may break this bond to create separate glucose and fructose molecules. When we have multiple glucose or monosaccharides together, we get a variable length chain called a polysaccharide. The main storage kinds that we see for plants and animals would be starch and glycogen respectively. So plants store their extra sugars produced as starch, animals store it as glycogen. We also see structural types of polysaccharides. The examples would be cellulose and chitin, found in plants and fungi respectively. The second category of organic compounds would be lipids. Now, lipids also contain carbon, hydrogen, and oxygen, but much lower proportion of oxygen. The 4 main kinds would be the neutral fats, phospholipids, steroids, and something called eicosanoids. We will focus on the first 3. The neutral fats, or triglycerides, have 3 fatty acid chains. These are variable length chains. These 3 dots here imply that we can have a variable length of chain. The chain is made of carbons bound together with different numbers of hydrogens. These fatty acid chains are then bound to a molecule called a glycerol. The bonding together occurs through dehydration synthesis, and the breaking of this molecule or triglyceride would occur through hydrolysis. The other type of lipid would be a phospholipid. Phospholipids are modified types of triglycerides where we see 2 fatty acid chains as before. However, we see something very different in this last group. Here we have a phosphorus containing end, right? This is polar. polarity comes from here having a plus charge on this nitrogen and a minus charge on this oxygen. So here we have a polar side and a nonpolar side in this molecule. These phospholipids are often depicted in a shorthand with a polar head shown here and a nonpolar tail. These will be extremely important in Chapter 3 when we talk about the organization of the cell, specifically the plasma membrane. Another kind of lipid are steroids. Steroids, one example would be cholesterol. Other types of steroids include things like estrogen, testosterone, progesterone. What we have here, these are lipids. These have a organization in which we see several carbon rings. The difference between, say, cholesterol and the other steroids, such as testosterone or estrogen, are the different chains and organization of those branching groups. All lipids have in common the fact that they do not readily dissolve in water. Some of the different locations where we might find lipids within the body are shown on this table. The triglycerides and phospholipids you would see in the fat deposits and in the cell membranes respectively. In the steroids, we see many different locations where we might find steroids. Many of these are going to be hormonal in nature, so estrogen and progesterone as well as testosterone are our sex hormones. Cortisol is what's called a glucocorticoid. This is an important type of hormone as well. The third group of organic compounds are the proteins. In order to understand proteins, we must look at the building blocks of proteins, which are the amino acids. All amino acids have a similar structure, in which they have an amino group and a carboxyl or a carboxylic acid group, hence the name amino acid. If we look at the structure, we have a central carbon. Here is our acid group. Here is our amine group. What separates all of the different amino acids from each other is shown here as an R group. That R group is a variable group, and it may be anything from a very simple R group in glycine, which is the simplest of the amino acids, to another acidic group. Here we have a CH2COOH. We have another acidic group, and this is aspartic acid. We can have very large R groups, and we can actually have R groups that contain other elements, in this case cysteine, which contains sulfur. In looking at amino acids, as we move into proteins, proteins are chains of amino acids, and the way that we see this are varying combinations of the 20 amino acids bound together with peptide bonds. When we look at any amino acid, ignoring the specifics by putting in an R for the variable group, right here we can see that from this acid side and this amino side, we have an OH and an H. Using dehydration synthesis, a bond called a peptide bond might be created here. Likewise, this molecule could also be broken down, and this peptide bond could be broken down through hydrolysis, the addition of water to create the separate amino acids. Now, proteins are going to be extremely variable. We will not go through too many specific examples because there is a near infinite number of possible combinations of proteins. So in order to understand what proteins look like, we will talk about the different structural levels of proteins. The basic structure is what's called the primary structure, and this is the amino acid sequence. That amino acid sequence is dictated by the DNA of particular cells or genes, which we will see in the later parts of Chapter 3. The secondary level will be what are called alpha helices or beta pleated sheets, and these would be specific interactions due to hydrogen bonds between near amino acids within that sequence. The tertiary level is going to be a three-dimensional or a superimposed folding of secondary structures. All proteins have some degree of primary, secondary, and a small amount of tertiary structure. Some proteins also have a quaternary structure, and this is where you have different polypeptide chains linking together to form a functional protein. To visually represent this, here we see a primary structure. This is a different way of looking at our initial amino acid. Here is our central carbon with the R group coming off. Here we would have our acid group, which is now bonding to the amine group of the next amino acid. These have a three-dimensional orientation, so you can see that the bonds are sticking up and out in different directions. Now, depending on what is going on with this organization, we will see hydrogen bonding. Remember hydrogen bonding from the previous chapter. Hydrogen bonds occur between hydrogens and electronegative molecules, or parts of molecules. So what we can see is we can see secondary structures either occurring in what's called a beta-pleated sheet, where we have hydrogen bonds forming between the oxygen and the hydrogens of parts of the molecule, or alpha helices, when we have a helix or a twist that occurs. Both the alpha helix and the beta-pleated sheet can then be folded to create a three-dimensional structure. That three-dimensional structure is due to interactions of the R groups. So if we have two R groups that are attracted to or repelled by one another, they will fold the overall structure. The fourth level then is when you have multiple subunits that must come together to form a functional protein. For this example shown, we have what is called hemoglobin, an extremely important protein in the transport of oxygen. And in order for this to function, it has a very particular three-dimensional shape and is composed of 4 smaller pieces that have to fit together in order for function to occur. Now, the different types of proteins, we have a main group of fibrous proteins in which the proteins are extended and strand-like. Here we find many of our structural or strength-inducing proteins, things like collagen, elastin, keratin, and spectrin. And you will see these in different places as we go through the physiology. These are also involved in movement, so we will see actin and myosin. We will look at these in depth in Chapter 9 when we talk about muscle contraction. When we have fibrous proteins, here we have more extended and strand-like proteins, as opposed to globular proteins. Here we have more compact or spherical shaped proteins with more higher level tertiary and quaternary structure. We see more diversity in terms of the function here. So we can see enzymes, the example previously shown of hemoglobin, many hormones that are peptide or protein in nature, as well as antibodies, are all considered to be globular proteins. The fourth category of organic compounds which we will discuss are the nucleic acids. Nucleic acids are actually quite complex. These contain carbon, oxygen, hydrogen, nitrogen, and now a new player, phosphorus. The basic structural unit of nucleic acids are the nucleotides. Here is an example of one particular nucleotide. A nucleotide contains a phosphate group, a sugar, and then this called a nitrogenous base, or an N-containing base. This particular one is a nitrogenous base called adenine, given with the A, so this is called an adenine nucleotide. Now, you have most commonly heard of nucleic acids when you talk about DNA and RNA. The difference between DNA and RNA is the sugar that's present, whether it is a deoxyribo or a ribo sugar. The different nitrogenous bases that we'd see would be adenine, given with an A, guanine G, cytosine C, thymine T, and uracil with a U. And as said before, the two main classes are DNA and RNA. DNA is going to be extremely important, and we will talk about its function in Chapter 3. But in an overview, DNA is a double-stranded molecule that exists in a helix that is found in the nucleus of the cell. This provides all of the instructions or blueprints for making proteins, so it gives the order for the primary level of proteins, so I mean the amino acid sequence. DNA must replicate itself before any cell divides, and this is the basic of our genetics. If we take a look at DNA, we can see that we have a phosphate group, a sugar, and a nitrogenous base. We also can look and see that we have a double strand. So these dots here represent hydrogen bonds. The hydrogen bonds do not create a new molecule, rather they hold a much larger molecule together. This shows you an adenine nucleotide, so here we have our A. This is thymine, this is T. When we look at a much larger example of DNA, we see that DNA forms this circular staircase, if you will, or this helical molecule. There's a certain relationship that occurs in DNA having to do with those hydrogen bonds. Okay. Adenine and thymine always form two hydrogen bonds. So A and T create two hydrogen bonds, whereas G and C, guanine and cytosine, form three hydrogen bonds. So A and T have two, whereas G and C have three. This purple ribbon here represents the sugar phosphate backbone, where we have a deoxyribose sugar bound to a phosphate, bound to a sugar, bound to a phosphate, bound to a sugar, bound to a phosphate, so on and so forth. Now, shown in this diagram, we have approximately 20 base pairs. In the human genome, there are approximately 6 times 10 to the 9th base pairs, which is somewhere in the realm of 6 billion base pairs. So obviously this is a very, very small part of our overall DNA. In addition to DNA, we also have RNA, which is ribonucleic acid. There are a few differences between DNA and RNA. Physically, the difference is that DNA is double-stranded, whereas RNAs occur single-stranded. RNA uses the nitrogenous base uracil instead of thymine. There are 3 main types of RNA that we will talk about as being important in Chapter 3, and these are messenger, or mRNA, transfer RNA, or tRNA, and then ribosomal RNA, or rRNA. And we will see these in depth in Chapter 3. Now, there's a very important type of nucleotide of RNA called ATP. And ATP is ubiquitous because it is a source of immediately usable energy throughout the cell. What we see in ATP is we see the adenine group, that is our A. Here is our ribose sugar, so we know that it is a type of ribonucleotide. The TP part stands for triphosphate. So here we see 3 phosphate groups. So instead of seeing bonding from a sugar to phosphate, sugar to phosphate, sugar to phosphate, instead we see bonds here. These bonds from ATP to ADP, diphosphate, and AMP, monophosphate, these are considered high energy bonds. So when a cell is taking energy in and storing it or using it, it will put it into ATP. Cells can then use ATP to do different things. When we are talking about active transport, ATP provides the energy through phosphorylation to change the shape of proteins to move substances against concentration gradients. When we look at muscle contraction in Chapter 9, ATP will provide the energy to do mechanical work and actually shorten the muscle. ATP will also be important in allowing for chemical work to occur by giving energy to reactants to create new products. So overall, ATP is a vastly important molecule, so make sure you have an understanding and you're able to recognize it.